most of you know me. And as one of the organizers of the C-DEBI Network Speaker Series, I am very pleased to introduce Dr. Julie Meyer, who's at the University of Florida. Julie's been doing wonderful work studying microbial life in cold, hydrologically active fluid at North Pond, which is in the North Atlantic, and it's been one of the principal project foci of C-DEBI. So I'm really looking forward very much to hearing what you have to say, Julie. Thank you for presenting to us.
Thank you very much. I'll go ahead and start up my presentation. So let me know if you can see that. Thank you everybody for tuning in. As Steve said, I'm going to talk about the cold oceanic crustal fluids at North Pond. And this is looking at both the chemistry and the microbiology. And this is work I did as a C-DEBI postdoc in the lab of Julie Huber. So where's North Pond for the folks of you that don't know? North Pond's on the western edge of the Mid-Atlantic Ridge, and it's a shallow sedimented basin. And you see from this bathymetry map here that it's surrounded by these ridges. And in this shallow depression, there's a relatively shallow layer of sediment up to 200 meters. And why do we care about some dip in the ocean floor in the middle of the Atlantic? Well, North Pond is likely to represent thousands of other similar basins like this along mid-Atlantic, mid-ocean ridges. And it's a place where we can study the cold crustal aquifer. So if we look at the schematic of cutaway of the crust, we see that most of us are familiar with hydrothermal vents, and this is where this red heated, highly altered water is being discharged into the ocean floor. But that water has to get into the aquifer from somewhere, and that's at places like the ridges surrounding North Pond. So seawater is able to percolate into the ocean crust through exposed basalt rocks. And in other areas of the ocean, most of the ocean floor is covered with sediments, and that seawater just can't penetrate through that layer, so it acts like a cap. So there are these areas where the seawater gets sucked down to the crust, and through low temperature hydrothermal circulation, it moves along across the through basalt rocks, tiny cracks and fissures in this unconsolidated porous rock. And as it travels through, it undergoes abiotic and biotic transformations. And we really don't know too much about what's going on there just because of the simple fact of limited access to it. So the way we do sample the crustal aquifer is through corks and boreholes. So the International Ocean Drilling Program has put lots of boreholes all over the ocean floor. Most of the time they're in collecting sediment cores, since most of the ocean floor is covered with thick sediments. At North Pond, we have a thin sediment layer, and we can bore down into the rock. And the advantage of boring somewhere like North Pond in this sediment basin is that we have that sediment to actually help anchor in that cork. So this is a reinforced hole, basically. It's got a platform on the top for ROVs and sampling equipment to land on and hook into the umbilicals that lead into fluid sampling lines down into the crustal aquifer. Sometimes we have instruments suspended inside the borehole, inside this cork, and this is sealed from direct interactions with the bottom seawater. And really the reason we want to get at this, it's like I said, there's transformations that happen in the aquifer, and because there's about 2% of the volume of the ocean is in the crustal aquifer, and these transformations happen, it does have an impact on global chemical cycling. So when we put these holes in the ocean floor, we do collect the rocks and the sediments that we take out of the holes, and these are put into repositories like the one in Bremen. This is a picture of there's the whole stacks and stacks of cores from the ocean floor, and these the core that's out on the table is actually one from North Pond. And so you get an idea of what this basalt rock looks like. And because we've been going to North Pond for quite a few years, we know a lot about the geology and something about the fluid flow at this area. We also know from looking at sediment cores that the aquifer below North Pond is oxygenated. So the deep seawater has oxygen, and the basalt hosted fluids also have oxygen, but the sediment basically is anoxic. So the oxygen is depleted very quickly in the shallow layers of the sediment. But as you go deeper and deeper, you notice that the oxygen concentration starts going up again as it interacts with the basalt basement. And we do know that there's some consumption of oxygen in the basalt crust, but not a whole lot. So we kind of wanted to have a better idea of what's going on there at North Pond. So here's a schematic of some of these cork observatories that we have at North Pond, and this is not to scale. But we do have 4 corks there. There are only 2 that I'm going to talk about today, which is 1382A and 1383C. Just give you an idea of what it looks like inside. 1382A, we have only 1 depth horizon where we can pull fluids from. But at 1383C, we actually have 3 different levels, and these are separated by these packers in between, and there are fluid delivery systems going to each level. And we don't know for sure how much vertical circulation is going on next to the boreholes, but within there we tried to collect fluids from different depths so that we get an idea of, are these, do things change as you go deeper within the crust? Because we do know that at some point the water and the rocks are going to heat up, and that has a big impact on the chemistry and the microbiology of the fluids. So like I said, I'm going to talk about 2 boreholes and 4 formation fluid samples. And these were collected in April and May of 2012. The boreholes themselves, the corks were installed in November of 2011. So these are fluid samples that came relatively soon after the observatories were installed. We also have deep seawater from about 4,400 meters depth. And so we looked at the chemistry and the microbiology of these fluid samples to get an idea of who's there and are they active and how many of them are there. So just looking at some basic chemistry from the different, I have here the depths of those layers. The pH is basically neutral, and there's not a difference between the seawater and the formation fluids. But with the oxygen levels, we did see a small depletion in the oxygen in the formation fluids. We also looked at a bunch of trace elements, and I'm not going to show all this big table full of numbers. But basically, most trace elements and dissolved volatiles were no different between the formation fluids and the bottom seawater, with a few exceptions. The exceptions were that uranium was depleted, and that's likely it's not unexpected. As the water reacts with rocks, the uranium gets depleted from the fluids. But we did see an enrichment in things like silica and cesium and barium, and those are likely reflecting interactions with the sediment pore water. So those are things that are common in sediment, and it's probably just diffusing into the crustal fluids from the sediment. So if we have a look at how many cells are there, we see that basically there's no difference between, again, the seawater and the formation fluids in the terms of how many cells are there. But we do see some reduction in the amount of bacteria versus archaea in seawater. So seawater had a little bit more archaea, but basically all of these fluids were dominated by bacteria. And the percentages I'm showing here are from fish probes with bacteria-specific probes. For archaea on those same samples, we just basically, you saw one cell every, half a dozen or a dozen fields of view. So it just really wasn't reliable, hardly anything there to count. We also confirmed this with quantitative PCR using both bacteria-specific and archaea-specific primers. And for the seawater, we could get some amplification, but basically for the formation fluids, it just wasn't consistent enough amplification using archaea primers. And this is agrees with actually what Jason said last week, or the last presentation about the old and cold crust. There aren't very many archaea there. It's mostly dominated by bacteria. And we also looked at 16S ribosomal genes to look at the taxonomic diversity of these fluids. And we used both bacterial-specific primers and archaeal-specific primers. And again, there just wasn't consistent amplification with the archaea. So we used FISH and quantitative PCR and regular V6, 16S PCR, and all three of them said there's really no archaea here, or not enough in the volumes that we filtered to be able to detect them consistently anyway. So if we look at the overall community structure based on their taxonomic identity, we do see that the formation fluids tend to cluster together, and this is looking at both RNA and DNA fractions. You see the two boreholes cluster together away from seawater, which is the deep seawater, and the mud there represents drilling mud, and this is it's minerals mixed with the surface seawater that's used during the drilling process. So we wanted to make sure that it wasn't the boreholes full of stuff that we introduced when we drilled. So if we look at the specific taxa that we detected, I'm just focusing on the major players here. So the black is things that were less than 1% in the samples, and the gray is less than 10% of the samples. Excuse me, less than 10% of the community. If we look at the most abundant groups, they're definitely different between seawater and mud and the formation fluid. So this is the first indication that we really have that there's something different going on in the formation fluids than in the bottom seawater, because the chemistry looks almost the same and the cell numbers look the same. But when we get to the actual taxonomy, we start to see differences. So some of the biggest players in the formation fluids are gamma proteobacteria and epsilon proteobacteria. So for the gammas, there's Caldwellia that is in all of the formation fluids, and this is a common group in deep sea habitats. But in this case, it was not very common in the seawater right above North Pond. It might be there in low amounts. We also saw the gamma proteobacteria, this Acidophorobacter, which only has one cultured representative of that genus, and it is an iron and sulfur oxidizer and a nitrogen fixer. So a likely autotroph, and in a basalt-hosted system, they would have access to iron, so that kind of makes sense. The other group that we saw a lot of are this Sulfuramonas in this kind of rusty red. And Sulfuramonas is an epsilon proteobacteria that's likely a hydrogen and sulfur oxidizer. And this is something that's common in low temperature hydrothermal settings like diffuse flow, places that straddle oxic and anoxic environments, like hydrothermal plumes where it's getting some kind of reduced chemicals from the plume, but it is still getting oxygen from seawater. We also see it in marine sediments, mats at hydrothermal vents. In this case, I would say the Sulfuramonas is likely coming from the sediment pore waters, because we do have some preliminary data from other folks that suggest that there is some Sulfuramonas in those sediments. So probably the Sulfuramonas is coming from pore waters. The Caldwellia is probably coming from the deep sea water, but just getting selected for in the formation fluids. And then this Acidophorobacter, it might be present in the bottom seawater, but it's likely something that likes basalt. So now if we go into the strain level of these dominant genera, we see that there is actually some indication that there's differences between the boreholes and differences with depth as we go into the crust. So looking at the dominant strains of Caldwellia, we can definitely see that there's clearly a different dominant strain in 1382A versus 1383C. And we also see that as we increase in depth at 1383C, that the dominant strain switches. So it gives some indication that there's differences between the layers and between the different boreholes. And you can see there's just almost no Caldwellia in the seawater and the drilling mud. A similar story is going on with sulfur ammonis. In this case, there's a dominant strain in 1382A and in the shallow parts of 1383C. But as you get deeper, there's a different strain of sulfur ammonis that's dominant. And one other thing that we should note is that in all of these cases, there's a higher proportion of sulfur ammonis in the RNA fraction than in the DNA. And so that indicates to us that this might be an active member of the community. The same with Caldwellia. There's a higher proportion in a lot of the RNA samples compared to DNA. The other thing it might indicate, especially in the case of sulfur ammonis, we have a sequenced genome from hydrothermal vents, and they have 4 ribosomal operons. So this might reflect that it just has more operons, ribosomal operons, more ribosomes per cell, and so it shows up more in the RNA fraction. But it also, like I said, might reflect activity. Finally, if we look at the strain level diversity in Acidophorobacter, we see it's basically the same dominant strain in all of the samples, but it seems a lot higher abundance in the very deepest horizon from 1383C. And again, there's a whole lot more of it in the RNA fraction than in the DNA fraction. So we also looked at activity. So are these bugs just hanging around and just sitting there, or are they actually active? Do they have the potential to be more active? So we incubated these fluids with labeled acetate to look at heterotrophic growth or potential rates of heterotrophy. And we incubated them at both 5 degrees Celsius and at 25 degrees Celsius. And we can see for the most part there's higher rates of activity in the formation fluids, and especially with the higher temperature in the deepest fraction of 1383C. We also looked at autotrophy by adding labeled bicarbonate to these same kinds of fluids, incubating them at 5 degrees and 25 degrees. And here we definitely see higher rates of autotrophic growth in the formation fluids, especially at when you heat up the deepest fluids. So given the differences between the dominant strains and those dominant genera, and the differences in the activity, it really looks like that there's something going on with depth. Things are changing, even if it's not reflected in the trace element chemistry in the formation fluids versus the bottom seawater. So overall, we see that there's a high connectivity between the bottom seawater and the crustal fluids. And that's not surprising, especially at North Pine. We know that the seawater is entering the crust somewhere nearby there. But we see this in the introduction of Caldwellia, and in the chemistry, there's just very little difference between the formation fluids and the bottom seawater. We also have indication that the sediment pore waters are things from the sediment pore water are influencing the crustal fluids, in that we see that diffusion of oxygen up into the sediment, as well as the diffusion down of things like the cesium and silica, and possibly the sulfur ammonis down into the crustal fluid. So you have this sandwich of oxic and anoxic habitats, but they all seem to be talking to each other, because we know that there's some oxygen penetration above and below. And we know that there's a connection between the bottom seawater and the crustal fluids through these recharge areas. So, like I said, we have an indication of taxa that are being enriched specifically in there, in the crustal fluids. So things that are getting pulled out of other environments, or maybe things like, or the acidifier might actually be a resident of the basalt. But they're definitely getting enriched in this environment. The contrast between RNA and DNA, especially in these dominant taxa, suggests that they are active. And the differences between the dominant strains at different levels and between the boreholes suggest that there's some kind of segregation between fluids at the top of the basalt layer versus further down. And we do have evidence for both autotrophy and heterotrophy. And we think that autotrophy plays a more important role in the crustal fluids versus the bottom seawater because they just have more access to electron acceptors like the iron. And that may be influencing who's present and how active they can be compared to the bottom seawater. In all cases, they're definitely limited by the amount of easy carbon to digest. So there's a potential for higher rates of autotrophy and heterotrophy, but they just are limited by the amount of carbon and dissolved organic matter that gets delivered to that system. So even though these fluids look a lot like seawater, there are subtle but important differences. We do see that reduction in oxygen and this change in dominant taxa, and this increase in activity, especially at the deepest level with the warm temperatures. So maybe that reflects there's some strains that are more adapted to the deeper levels as the crust gets warmer, or maybe it's just a kinetic effect, that they can just be more efficient at their metabolism at these warmer temperatures. And this study I'm presenting is the fluids that we collected in the spring of 2012. But when we were there, we left down this sled that collects additional samples, and that was just recently picked up at the beginning of this summer. This is an actual photo and not a schematic of the cork at 1383C, because it's got the 3 umbilicals. So this is the actual cork. This is the sled that's sitting on top. I guess it's gone now, but it was the sled that we deployed. It's got batteries, and basically the pumps are scheduled to turn on at certain times and pull up more water and filter it and preserve it, the filters with RNA later. And these have been retrieved now. So they collected several different samples in the intervening 2 years, and more samples of the water fluid, of the water. The formation fluids were also collected at the beginning of the summer. So this is just the beginning of a story. As I said, the fluids were collected only 6 months after the borehole was drilled and the cork installed. So it'll be interesting to see, is this a stable community, or is this something that's going to change over time? And of course, I want to acknowledge all of our collaborators on this project for all the chemistry, and also Beth Orcutt for the sample of the drilling mud from the installation of the cork. And of course, to the logistical support for putting in these cork observatories and collecting samples from the bottom of the ocean. And with that, I guess I can turn on my video and take any questions. Okay, I do see one question here about the percentage of fish stainable versus non-stained cells. Well, we definitely used fish, and Uli did that. So I'm not sure how many of the cells weren't stainable. For the cell counts, I think we did about 40 mils, 40 mils maybe. We do know that they're low. For the DNA extractions, we had filtered 5 and 10 liters of formation fluids. The Propinium bacterium, I don't know how different the sequence for that is from skin flora. We're looking at the V6 region, so there's only 80 base pairs there. So I don't know if we have anything representative of that in environmental samples or not. But I was kind of nervous about seeing that too. I don't have any measurements of organic matter. I think those things are being analyzed by other folks, though, the carbon. The iron, I don't think anyone measured dissolved iron in the fluids, but we do know that the iron is available, in the rock. So we wouldn't necessarily be able to detect it in the fluids, but it would still be available to microbes in the fluids that are passing by. And one important point is that there might be a lot of things in the aquifer that are actually attached as biofilms to these crustal rocks, the basalt rocks, and we wouldn't necessarily pick them up consistently, at least in the fluid samples. And we do have other groups looking at the actual rock from North Pond. I think we only saw sulfur ammonis. I don't think we had any sulfur ovum. So for the nitrogen fixer, the acidophorobacter might be a nitrogen fixer. Like I said, there's only one cultured representative of the whole genus. And I can't remember where it was from, but it wasn't from a marine habitat. So we don't know for sure that that's what it's doing there, but the acidophorobacter might be a nitrogen fixer. As far as staying place in this fluid flow, it's probably, I mean, it's not like I don't know what the fluid flow rate is, but it might not be that forceful for an individual microbe to experience, especially through some of the smaller crevices in the rocks. But I would assume that there's probably a lot more, I think there are probably a lot of things that we're missing that are attached to the rocks. Especially, we know that there's a big difference between things that are attached to particles in the deep sea water column versus what's free living. So I would imagine that they're trying to be attached to those rocks if they're depending on them for redox reactions. The manganese cycling, We didn't really look for that. I mean, I would suspect there are a lot of things that are down there that just aren't well characterized. And so we don't know which groups might be doing that. Um, I don't think anybody's looking at the fungus from the fluids. But based on what they look like, I mean, just what the filters look like, the fluids look like filtered on a DAPI filter, I didn't see a lot of eukaryotic cells. So for the incubation times, they were actually relatively short, but I don't recall the incubation time offhand. We had other long-term incubations that weren't labeled the first time around, but I know that there are a lot more incubation experiments for the most recently collected samples. But yes, the autotrophic activity was greater than the heterotrophic activity in our initial incubations.
All right. No, we still have some more people typing. All right, thank you, Julie.
Yeah,
So,
thank you, everybody, for listening.
yeah, so stay tuned for the next network speaker series. It's going to be in October with Sean Jungbluth, and we'll be posting this the video of this presentation online in a week or so for anyone who missed it. Thank you, Julie.
All right, thank you.